Hi, everyone. This is your People Behind the Science podcast host, Dr. Marie McNeely, and I am excited to be chatting about life and science with another great guest today. This is episode 637, and we are joined by Dr. Sandra Encalada. Listeners, if you've been enjoying the People Behind the Science podcast, we would be really grateful if you could share our show with a friend or tell the world what you think about our program with a review on your favorite podcast platform. Now, we have another great conversation on the way from today's guests. So listeners, get ready to meet another one of our phenomenal people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to the People Behind the Science podcast. Today, I am thrilled to be speaking with our guest scientist, Dr. Sandra Encalada. So, Sandra, welcome to our show today. How are you?
I'm doing great. Thank you so much. It's awesome to be here.
Well, we are excited to learn more about you and more about the work that you do in our conversation today. But let me start by just giving our listeners a little bit of background about you first. Listeners, Sandra is the Arlene and Arnold Goldstein Associate Professor in the Department of Molecular Medicine, as well as a Dorris Neuroscience Center Investigator at Scripps Research Institute in California. She received her bachelor's degree in physics from Earlham College, her master's degree in population genetics from the University of Florida, and her Ph.D. in molecular genetics from the University of Oregon. Afterwards, Sandra was awarded a Damon Runyon Cancer Research Foundation Postdoctoral Fellowship, where she conducted research at the University of California, San Diego. Next, Sandra worked as an assistant project scientist and Neuroplasticity of Aging Fellow at the University of California before joining the faculty there at Scripps. She has received numerous awards and honors over the course of her career, including the Glenn Foundation's Glenn Award for Research in Biological Mechanisms of Aging, the Baxter Foundation Young Faculty Award, the Ellison Medical Foundation New Scholar in Aging Award, and others. And today, Sandra, we're excited to get to know you both as a scientist, but also as a person. So can you start by telling us what you like to do when you're not doing science?
I'm doing science quite a bit, I got to say, but I lately spend quite a bit of time with my 5-year-old girl. During the time that I have been afforded the opportunity to be working at home, it's been a good time to bridge good bonds with her as she's transitioning in these early stages of her life. So I spend a lot of time with her reading and doing a lot of fun games. But in addition to that, when I manage, I'd like to swim in the ocean. I like to go out. We're here in San Diego, and we're fortunate that we can swim year round with a nice, good wetsuit, of course. The water is pretty chilly in the Pacific, but swimming in the ocean has been really just a wonderful outlet for letting go of a whole lot of thoughts of science that we scientists live with pretty much 24/7 otherwise. So I love swimming in the ocean and the camaraderie that comes with being with my pod, as we call ourselves. But in addition to that, I regained back my interest in music and in piano. I was classically trained when I was a kid growing up in Ecuador, and I've taken up piano again. I finally was able to manage buying a piano after almost 30 years of not having one. But now I've taken up the challenge to do so by learning how to improvise. And it's a very different mindset than doing classical music, but I'm really enjoying doing that.
Oh, that is so cool. So when you improvise, is it sort of like jazz style, or do you stay in the classical style?
Jazz, exactly.
Oh, cool.
So that's been a big challenge for a classically trained brain, So it's been really great to learn the chords and all these progressions and all that. It's definitely a different way of thinking, but very welcomed at this point.
Oh, that is so cool. Well, it sounds like you've got quite a bit to keep you busy outside the lab. And I think that change of scenery, like you mentioned, always helps. But I want to talk about your science as well today. So, Sandra, how do you describe what you do in the lab to someone who's maybe not familiar with their specific field or your research?
So we are what's called cell biologists. And what that means is we like to look inside cells to the little smaller parts that cells are made up, specifically brain cells, also called neurons. And these neurons are really specialized cells that are all over our bodies. But one of the features that are really fascinating about the cells is that they're so long and they're really, really polarized. They have these really long projections where you have the main body of the cell, it's called the cell body, could be almost a meter away from the very tip of it. So they have these really long projections of fibers that are called axons. One of them, for example, goes all the way from our spinal cord to the tip of our toes. Those are very, very long projections. And so what my lab studies is how things that are made in the spine, in that cell body, travel all the way down for about a meter in length to the tip of that nerve fiber in order to transmit information, both at the end, but also all along that nerve fiber. And that process is called transport or trafficking. And it's absolutely crucial for that neuron to perform the function that it needs to perform, right? So to transmit signals, to communicate between other neurons, to form memories, if it's a neuron in the brain. And that process of transport is highly regulated. And it's actually done by this fascinating set of machines that are called molecular motors. They're this little nanomachines that are literally bipedal in some cases. And they grab different things that are like proteins that are really important for the cell that are made in the cell body. And they take them along the axon. They walk them along the axon on these sets of railroad tracks that are lined up all along the axon that are called microtubules. And this process is really, really highly regulated and has to be very orchestrated to allow these machines to deposit all these components all along this meter length, in some cases, nerve fibers, so that the neuron can function properly. So my lab is really interested in how this process is accomplished in healthy cells. But it turns out evidence from many labs also suggests that this process is highly disrupted when we encounter neurodegeneration. So my lab also is interested in what fails about this transport process. And one of the things that we see is there's traffic jams along this really long nerve fiber that are very related to neurodegenerative symptoms and neurodegenerative diseases. And so we build models in the lab to study how those traffic jams are formed and what the consequences are for the health of the neuron and eventually for the health of the whole brain and the symptoms that it produces in people.
Oh, well, this is fascinating. And I love that metaphor that you kind of helped us envision these traffic jams along the axons. I think that's really helpful for people to conceptualize your work. And I'm sure you can tell I get excited just talking about the nervous system. I'm a bit of a neuro nerd myself. So it doesn't take too much to get me interested in this kind of work. But I know we all have some days where you could use a little extra motivation or inspiration. Some of our listeners might be having that kind of a day today. So, Sandra, do you have a favorite quote or a saying or something that just really motivates and inspires you?
One of my favorite motivational quotes is from Ruth Bader Ginsburg. And she said, "Fight for the things that you care about, but do it in a way that will lead others to join you." So I like this quote a lot because it makes me think about how it applies to so many areas of life, right? The obvious ones in your personal life, but also in science. So certainly in everyday life, Ruth Bader Ginsburg achieved such mastery at persuading people about things that she really cared about. For example, fighting for women's rights in so many contexts as she did. But for me, this quote is really important in this way, but also it applies to how we do science and our scientific enterprise, right? Because to me, a way to lead others to join you in science and being persuasive at doing so means to ask meaningful questions and then do the best and most rigorous job that you can at answering those questions, right? So design the best experiments that you can. And I think that's the best way that you can have others be persuaded by the science you do, is use your imagination to design ways to test hypotheses and then use your expertise to go for the experiments to get those answers, right? And then, of course, when we don't get the answers that we thought we might, it's just likely because there's something very interesting happening instead. But going for that persuasiveness in how you go about doing science and the rigor of science, I think is something that follows directly from that quote by Ruth Bader Ginsburg.
Oh, I love it. And I think Ruth Bader Ginsburg is certainly an inspirational figure for many people. And I think having these people in your mind as you encounter challenges, even as you just go through the everyday mundane situations as a scientist, can be helpful. So for you, Sandra, when you think back about your career, whether it was your early days or even just yesterday, are there people who really inspired you or kind of helped shape you and become the person that you are today?
Indeed, a very influential role model for me has been for a long time Barbara McClintock. For our listeners who might not be so familiar with who she was, Barbara McClintock was an American geneticist who made one of the most important discoveries of the 20th century, So until her work, genes were considered to be pretty stable units. They were arranged along chromosomes like beads on a string. But in the late 1940s, Barbara discovered that genes could be mobile and they could jump around and insert themselves in new locations in chromosomes, thus bringing about regulatory functions to those new places where they inserted that were unseen and unheard of before. So this now very well established principle of this jumping genes of transposons, as they were called, was an incredible challenge back then in the 1940s when she discovered them. It challenged basic tenets of science at the time. And really, people, as she quoted, said, they thought that I was crazy, that I was absolutely mad. So the discovery itself was very inspirational, and it had so many consequences, right? So it turns out about 45% of the human genome has these jumping genes. So we really know and are right now discovering how they affect so many different parts of biology, including neurodegeneration, which my lab studies. But however, one of the most inspirational things in thinking of Barbara McClintock has been how she went about her science and her life. Her work was so seminal. It took about 40 years to be really recognized. And of course, she won the Nobel Prize for this recognition. But how she went about it was amazing because she worked in a genetic system. It was maize or corn. And she did this by breeding corn strains patiently and painstakingly and following their traits for over many, many generations, right? At a time where it was a confluency of science taking different directions, but also when a woman like Barbara was not really featured and was pretty much left alone to do her science. I think eventually that worked amazingly in her favor, which is incredible how long she worked. In fact, she won the Nobel Prize alone, the first woman to win the Nobel Prize on her own. But how she went about it with the observation of her science, of her strains of corn. In fact, Evelyn Fox Keller wrote a beautiful biography on her where she tells about how she would sit with the organism that she was studying, in her case, the corn, and just study it and have a feeling for this organism. And this is something that has really made a big mark in my own enterprise of doing science, because one of the things that I think was really important is observation, the power of observation, and to sit like Barbara did for so many years with that organism, to really become one almost with it in terms of understanding it from every angle. That feeling that you get can impart a whole lot of new ideas and ways of thinking about the system that you're studying that is not necessarily very apparent if you don't spend that time with the organism. So Barbara has been really inspirational and an example of persistence in science that I go to in my mind very often, especially during difficult times in science, which there are plenty.
Certainly. And I think even just thinking back historically, we've had so many advances in science, and particularly in genetics. It's difficult to imagine how people can make these amazing, groundbreaking discoveries with just the limited tools that they had at the time.
Indeed. It's a labor of love in many, many ways, as well as endurance. Certainly at that point, it was very basic, like you said, with just crossing strains and looking at chromosomes under the microscope. This in part is such an amazing discovery. So it is something that I look back on, and I tell my students a lot about how to go about thinking about doing science.
Oh, absolutely. Well, Sandra, as you tell these stories, I think your passion for science really comes through here, but I'm curious to hear a little bit more about where it all began for you. So do you remember these earliest memories of yours when you first started getting interested in science?
So I was born in Ecuador in South America, and I was the first of 4 girls that grew up in a situation where we were very encouraged by both my mother and especially my father to think about science seriously from the very early on age. And I know people sometimes relate to when you were about 6 years old, and I remember being about that age and just wanting to kind of find out how things work. And I didn't break so much toys and open them up, but I like to plug things around walls and look at how things move around. And so I unfortunately ended up, breaking one of my father's early radios because I wanted to make it sound better. And I plugged it, instead of to the speaker, I plugged it to the wall, and the whole thing blew up. But my inquiry about how things work definitely was there. And the earliest memories that I have of wanting to discover things. But there were special points in time that I think made a big difference in my life in terms of thinking about science. And one of the earlier ones also was when I was about 12 or so. My father at the dinner table used to ask us, what do you want to do? And I said, I think I just like to think about how things work, but I'm not sure how to go about it. And he said, I think you should really think about studying physics and math because it explains a lot of things. And when you know about math, you can start quantitating things and counting things and making sense of that, how things work in a quantitative way. And you need to be taught math rather than learn it on your own. So study math first, and then you can learn a lot of other things if you really want to later, like history and other things that are easier understood and studied and learned from books. But math really needs to be taught. And I took that to heart, and I started earlier with studying math and physics when I was in high school, because in Ecuador, you had to kind of choose different areas and fields when you're young, actually. And I developed this love for physics that I carried on. Later on, when I was about 15, I had an opportunity to apply for a fellowship to come to the U.S. to study in an international school to finish high school, but it was called the United World Colleges. And I was in New Mexico for 2 years. I came by myself when I was 16. And the idea was to spend 2 years in this college where there were 200 students from about 80 countries back then in this sort of international environment. And then I would go to Ecuador. But then after that, I applied for college here in the U.S. and I got a full ride to go to Earlham, where I studied physics initially and was going about liking physics quite a bit, except that I started taking some biology courses at the last couple of years there. And I attended a seminar by this former alumnus who had her own lab and who was studying these things that she called microtubules inside cells, which are these little railroad tracks that I had mentioned earlier. We now study where these molecular motors carry things along when they grab them to position them from one part of the cell to a different part. And she had this slide that she had us look into where these microtubules were labeled with a fluorescent protein and line up all along like these beautiful asters inside the cell. And once I looked at that cell, I said, you know what? I want to be looking at that for the rest of a whole lot of my life. Mm-hmm. And amazingly enough, we're still doing that. And I was fortunate to be able to follow that dream. And I switched from physics to biology. I took a little long ways to get back to looking at these little railroad tracks. I did a master's at the University of Florida studying population genetics of sea turtles, out of all things, which provided me with a beginning interest in genetics. And then that took me to Oregon, where I really got enamored with studying these early processes where microtubules are absolutely critical in cell division. So I joined Bruce Bowerman's lab there to study the very first cell division after this nematode called C. elegans divides from a one-cell embryo to a two-cell embryo. And that very first division is absolutely dependent on these microtubules that I had been so enamored with when I was an undergrad at Earlham. And so I studied that for my Ph.D. and how that occurs. And that brought me then later to think about where else these processes inside cells, like the cytoskeleton, which is what these microtubules are, is the skeleton of the cell, because they are actually very important structurally to maintain the structure of the cell. There's long rods that keep the structure of the cell intact, and how they could be making an impact in the brain cells and neurons. And it turns out, as I mentioned a little earlier, they're absolutely crucial for maintaining both the integrity of these really long fibrils that line up so much of our body, but also for communication between different parts of this one really long cell. So with that, from Oregon, I came to the University of California, San Diego, to the lab of Larry Goldstein, where I set up a little bit of a project that was unique to the lab. I started working on what happens with these long cells when you challenge them with neurodegenerative stresses. For example, when you mutate a protein that we know confers a neurodegeneration disease. And in particular, I started looking at prion diseases. Prion diseases, for example, mad cow disease is one of those, and in humans is Creutzfeldt-Jakob disease. Those are diseases that we know are largely affecting neurons, and neurons die in these diseases by mechanisms that were still really unknown. And one of the very exciting things that I started investigating at UCSD during my postdoc was how is it that the cytoskeleton could be contributing to the death of this really long fibrils called axons. So that's what brought me there. And then I was fortunate when I was applying for faculty positions to have the opportunity to stay in San Diego, just across the road at the Scripps Research Institute, where I started my lab about 10 years ago. And we have continued doing this work on looking at this amazing fibrils, microtubules inside neurons and how they could affect disease.
Oh, that is wonderful. And I think, Sandra, your work is absolutely fascinating. So I'd love to go in a little bit deeper if we can. So can you maybe tell us a little bit more about one of these projects that you have going on right now that you are just so excited about?
We're very excited about what I consider perhaps one of the greatest contributions of my lab to date, which is in this fibrils again, going back to this axons. One of the observations that has been made for decades now is that the axon is one of the first things that suffers in people that have, for example, Alzheimer's disease, Parkinson's disease, Huntington's disease, you name it. It's one of the first vulnerable parts of these neurons that die, while the soma seems to have some more resilience. And one of the features alongside with that has been that axons have and develop in brains of people with dementia and with all these neurodegenerative diseases. They develop these really large swellings that happen along the axons, like beads on a string. The axon swells up very large. And inside of those swellings, if you take a look at it, there's accumulations of all kinds of things that seem to have piled up there and gotten stuck, a bit like these traffic jams, like I was mentioning to you earlier. In fact, that's how we call them. And in addition to that, there's aggregation that's happening there. There's proteins that are involved and that are very crucial and involved, and sometimes they're mutated in people that have neurodegenerative diseases. For example, in the case of the prion protein, prion diseases are different flavors of this disease. They also have these axonal swellings, like I mentioned, but they're caused by sometimes mutations in this protein that's called the prion protein that you can inherit from your parents if one of them has a copy of it. And these diseases are similar in some ways to Alzheimer's in the sense that they're manifested as dementias, but there's lots of movement disorders for which there is no treatment or cure now. Unlike Alzheimer's, they're very fulminant, so they occur very fast from the onset of symptoms, and they're fatal within a year, usually, of this onset. So in these axons, when going back to the swellings, what has been observed is that the swellings happen really early on in disease, in both humans as well as in models of disease that people have built to study prion diseases as well as Alzheimer's disease. And I was very struck earlier in my career as how early they come. So I was thinking, what do we know about how they're formed? They might have consequences, obviously, to the cell. And lo and behold, there was nothing in information in the literature as to how they're formed. So when I set my lab about 10 years ago, we set to model this swellings along axons and to try to identify pathways that are accounting for their formation. And so this is the story that we have painstakingly worked on for a very long time, and that I'm proud to say is coming up pretty soon in print, where we've basically discovered a set of genes and proteins that very much depend on that movement that I was mentioning by these little nanomachines that walk along these microtubules. And they take, it turns out, a mutant form of this prion protein when we put it inside neurons. They take it from the cell body and they take it all the way along the axon. And once they get in this long fibril in the axon while they're walking along, they take them inside this sac-like filled vesicles. And these vesicles, they encounter each other along the axon and they start merging with each other. And when they merge, inside is that prion protein that has a very, very strong affinity to form aggregates if it's put together with another of its kind. So you start forming this really large sacs of aggregates of this disease and pathological form of this prion protein that then starts growing in size. And this is what turns out makes this distensions or swollen axons along all the length of this really long axonal fiber. So when we discovered these proteins, of course, the way that we discovered that they were involved in the formation of these very large aggregates inside the sacs of vesicular sacs is because we reduced their function in these neurons that we were growing on petri dishes in the lab. And when we reduced their function, these aggregates were almost completely gone. And we showed that when they were completely gone, they actually restored the function of these neurons, which was very affected when the aggregates are there. And the neurons live longer. This was very exciting because it told us a lot about the mechanisms of what happens in the requirements of this process of movement that happens inside neurons and the very basic biology of this trafficking process. But it also allowed us to discover potential targets for this process. And indeed, we're very excited because we've followed up this work that is being published very soon with more in vivo work that is working in actually whole organism, in this case in the mouse, where we've built models where we're finding that this works just as well as it works in this neurons in these dishes in the lab. And so we're very, very excited about following up this work in real organisms and also in discovering potential molecules that could be doing what the genes in these proteins are doing now. Without us having to manipulate directly those components that we found, we can use molecules to also disrupt the function and the growth of these aggregates and hopefully restore function within the context of this very devastating prion diseases.
That is so cool. Sandra, listeners, we are going to have to keep an eye out for this and hopefully other papers coming out soon from your group that go into all the detail of these projects. And I think it's great, of course, to be able to work on really exciting research questions and projects in the lab. But I think every scientist has days where it just seems like everything's going wrong. And so far, we've only talked about the rosy moments. But Sandra, can you tell us about a time when you really struggled with something, or maybe you had a major failure that you had to work through, and tell us how you did it?
Just circling back to the story that I just mentioned now, this discovery that we made in terms of this pathway, I think it took a while to get to this point where we could put together the story, and it came about with a few setbacks. One of the earlier ones that I think is worth mentioning because we really learned from it. And basically, a terrific postdoctoral fellow who really spearheaded this project. His name is Ramon Chassafer. He's no longer in the lab, and now he has his own very successful career in biotech. But he was working on this project and stumbled onto this result that movement out of these particles along these microtubules was out to the axon was really critical to make them. Like if you don't have that movement, you simply don't take this misfolded prion proteins out to the axon, and then you don't have aggregates. You don't have the prime material out there in the axon to make those aggregates. However, that result was completely in contradiction to a very established tenet regarding how aggregates might form inside neurons. Previously, a whole 2 decades of very, very good, important work, including my previous postdoctoral fellow advisor's work, has shown that when you remove that movement of important misfolded proteins to the axon, you make things worse, not make things better. And we were finding completely the opposite. So we stumbled onto this data, and I told my postdoctoral fellow, first thing, of course, that a faculty member says is, how many times have you repeated the experiment and how many cells have you looked in? Mm-hmm. And he said, I've done it about 3 times in about 50 cells. And I said, yeah, that's pretty good, but you know what? Let's do it again. Right. And so he came about a week later with like 150 movies. About 3 more times he had done it. And it was very, very clear that this is what's happening. And then I said, well, now we have an issue in our hands. We got to explain this to a field that has seen otherwise. That would made for a very interesting few months of us really doubling down on trying to figure out how it is. And, the end of it and the long of it was that we were looking at really early stages when things were happening, where this process of moving things out of the soma into this long axonal projection was absolutely critical to make those aggregates. But once you make them, and you spend a long time making them, then removing the movement, the process of moving things around could make things worse. And the reason why people had seen this at later time points in all of the models that they had studied previously, well, we went really early in that event. So that's how we can very easily actually reconcile these two. But what it did is actually it set us back a little bit in terms of science, but also trying to publish this work and also trying to write grants based on this sort of newer idea. I got to say, I'm coming through out of that period of time where we're getting there, but it's been a bit of a going back to Ruth Bader Ginsburg, persuading with the best science you have, and in the ways that people would join you. And so we had to endure quite a bit of criticism and of concept building here that I think it provided inspiration and motivation to continue, because when you are sure about your data, you just kind of keep going, right? But I think that was a bit of a hard time and a hard road of learning experience.
Definitely. Well, Sandra, thank you for walking us through this difficult time. And I really love these stories of unexpected findings and what it takes to uncover the truth of what's going on when these unexpected findings pop up. So those are wonderful. And in this case, of course, it led to a success. And we love sharing the stories of success of scientists. So do you have a favorite success story that you'd like to share with us, or even just a small win recently?
I think that getting this work out and being recognized both with a paper and I think a grant as well, that turns out just recently is a success story. It's been a long road with this work. And I think I measure the success both, of course, in the tangibles. But the amazing thing is just to circle back to the people who've done this work. And to me, both seeing the pride that they carry and having been part of the story is a big part of it, but also what they take from it as they move forward in their own careers. I think that has been, to me, a measure of success that is less tangible maybe immediately than having a paper published or grant funded, but is what I can take a look at and see as part of my legacy and the legacy of the science. And so I think that's the success, is celebrating how this contributes to the amazing people that worked on this in their careers.
Oh, that is wonderful. And I know a lot of hard work from a lot of people goes into making these projects a success, particularly these ones that span over many years. So do you have a favorite way that your lab celebrates?
When these things occur, we always celebrate by getting together and uncorking a little bubbly to try to celebrate that. I have this little tradition that when we publish a paper, I print the first half a page or so, a quarter of a page in a mug, and I give it to all the authors of the paper. And in this case, we're also getting our cover in published. So I think I'm going to put that as well there in mug. And so most importantly, we just, acknowledge together how this has been a wonderful long road, but a good learning experience. And, our contribution to science is really how we're celebrating and moving forward to potential discovery of therapies, which is actually a very good reality in this case.
Definitely. And I love this idea that you have of almost like a little trophy case of mugs that people can have and remember all of the work that they did. So that's phenomenal.
We're excited about those. Everybody's waiting for them. I still have to get this one printed for this latest work.
Oh, that is phenomenal. And I know we've talked a lot about the research, and I know it requires a lot of reading. You've got to stay on top of all the new information that's constantly going out there in the field. So I always encourage our listeners out there to maybe take a step back, do a little reading for fun, let your mind wander a little. So, Sandra, do you have a favorite book that you can recommend for our listeners who might be looking for their next great read?
So my latest read is by Konnikova. It's her book called The Biggest Bluff, and I loved it. She takes us in this book into her really remarkable journey through learning how to become a poker player, like a professional poker player, from being a complete novice and not even knowing how many cards are in a deck of cards. Her writing is really vivid. She takes us right into the poker room with the sounds, the smells, the visuals, and she puts us right there on the poker table alongside her competitors. But importantly, she's a psychologist and she writes about social psychology. And this book is really about how people make decisions and whether our lives are shaped by decisions or by chance. Of course, in poker, this is everything, both decisions and chance. And how do you up your odds in the game? One of the things that the book stresses is failure being such an important process in this both decision making and just influencing chance. So one of my favorite quotes from the book is, the benefit of failure is an objectivity that success simply can't offer. And to me, this is really, really true, right? She highlights how learning how to do anything, how to think, how to cook, how to write poems or anything else, the only way of really learning how to do those things is failing. When you're first doing and learning how to do something, you'll have absolutely no way to gauge if you're really just that brilliant or if it was a total fluke and you just got incredibly lucky, right? So this again brings me back to where I think innovation and discovery happens in science, right? And it often happens when you fail.
Well, excellent recommendation. I have not heard of this book, so I'm going to have to pick it up myself, listeners. The Biggest Bluff. We'll have to put it on our lists. So Sandra, thank you so much for that recommendation. And I know we've talked about some of your experiences in science. And I think one of the things that's really exciting for scientists are some of the opportunities to travel and to talk to people all over the world. And I know, of course, the past few years have looked different for scientific travel. For you, is there a particular place that your science has taken you that was just the most memorable that you can tell us about?
It was a trip that I made to Cuba. It was back in the 1990s, and I was a master's student studying population genetics of sea turtles at the University of Florida, and I was an Ecuadorian citizen, and my advisor then had been invited to give a talk there because Cuba was very interested in protecting these very endangered sea turtles there. So he asked me to go because I could travel there at that point and give a talk in Spanish, which I was happy to do. And going there was really an eye-opening experience at that time. I was a young student and I went there with a mission, which was to give this talk and represent our work that we had been doing. But what I ended up taking, in addition to the science, is the amazing resilience of the Cuban scientists and people in doing research, but also in their humanity. Because it turns out, and this is a small anecdote, when I went there, we were there for about 5 days and I had taken enough money to pay for my hotel room, basically, to spend there. And it turns out, because of a lack of communication before we traveled there, I only took half of the money that I needed to be there. And so I ran out of money. And so of course, I was a student and I whipped out my credit card, my Visa, and I said, okay, here, I pay for this. And they said, well, we don't take Visa here. And so I ended up running out of money. And one of the people there that they had assigned to us as hosts invited me to stay with her family there. And I spent the next 3 days just reveling in the amazing, like I said, resiliency of the Cuban people and resourcefulness and how they go about, in the case of the scientists, living and trying to do science in an environment that was resource limited in many ways. But the humanity of these people and just taking me in and just opening their hearts and home, even with the very limited resources they had, was really amazing. And to experience that was great.
Absolutely. And I think you find in meeting colleagues from all over the world that there's sort of something that brings you together, just being scientists in the field, being excited about the same things. And everyone does seem to be, in many cases, really welcoming. So I'm glad you had this experience. And it sounds like they saved the day in what could have been a really difficult and stressful situation.
It sure did.
Well, I love talking about the great people that you get to meet in science and work with. And I think you've hinted a couple times through these stories at some of the great people that you've met. But I think that's not always how scientists are portrayed in the media, unfortunately. So there are still some stereotypes out there. And we try to break those on our show by talking about these fun, funny, maybe human moments in science that people maybe don't always get to hear about when they see scientists in the media. So do you have a story of yours, Sandra, that really captures this human side of science, whether it's a quirky tradition or a funny story or memory?
I do. I have a funny story. This actually relates to just within the last 3 months or so, our very dear colleague and friend, Artem Pataputian, who won the Nobel Prize about 3 months ago. And he's in our institute and we're part of the same neuroscience center. So his lab is just above our lab. And basically, after winning the Nobel Prize, of course, there was a lot of busyness around that with the media and all that. And Artem really decided to include the small group of faculty that are in the center, in this neuroscience center, in many of the festivities, because this year, because of COVID, there was a lot of cancellations and he couldn't go to Stockholm. So he actually invited us to a few of these events that happened, including the ceremony for giving him the medal. So that was terrific, which happened here at UC Irvine at the National Academy of Sciences building there. But, Artem actually it's really just to show how great minds could also be amazingly funny people, right? He has been sort of known for being a bit of a prankster with us. And I had always been in the gullible receiving end of that, unfortunately. And previously he had sent emails in years past with kind of April Fool's jokes. And I had been, like I said, gullible. And so after about a month after he won the Nobel Prize, I got this invitation in the email to attend a ceremony that the Embassy of Lebanon, where Artem is from, was going to confer him one of the highest honors as a Medal of Lebanon, and to RSVP to the invitation that I was invited to attend this at our campus. And I thought, well, this is great. Artem is not going to get me this time. So I didn't RSVP, but what I did is I wrote back to Artem and I said, hey, thank you for the invitation. I'm really impressed at your Adobe Illustrator abilities to send me this really nice electronic invitation. But, I did RSVP. Then I told them anything short of a written invitation and written in gold and plated with a couple of diamond rings, I will not be able to accept their invitation otherwise. And he wrote me immediately. He said, oh my God, I hope you did not send this because this is for real. And I told him, oh, I'm really sorry, but I already sent this response. And then there was silence for about 5 minutes. And I just sat there in my office, not getting any email back. And I ran up to his office just to explain this whole thing was a joke. And he was bent over, just laughing he was laughing so hard, he had almost tears in his eyes because he thought it was just the funniest thing. And so, of course, I told him that I'd never RSVP'd and that I will RSVP for real, but that there was a hint of doubt that this might be real. So, and he appreciated that. So anyway, it was a good laugh that we had.
Oh, that is wonderful. Yeah, I think having that sense of humor is so important in science. And there are so many funny, amazing scientists that I've met myself. So this is a phenomenal story. And it sounds like you're able to answer so many really cool questions. You're working in a really great environment with brilliant colleagues. And oftentimes, even in these situations, you face barriers, whether it's technology, whether it's funding, limited staff, feasibility. So you're not able to answer all the questions you'd like. But if we took away all of the restrictions for you today, Sandra, is there one question in science that you would want to tackle most?
A thing that fascinates me is the link between viruses and dementia. I think this is really, really important. I think that, this might sound opportunistic now that we're living through this viral pandemic, but the link between viruses and dementia has been there actually for a while, but it has been a bit controversial as to really, is there a link in the first place, which I believe that there's evidence for, but how this could be happening, right? How viruses might be affecting our brain and maybe leading to dementia. And this is really, of course, a timely question now, not just with this exogenous viruses that now we have to battle, but also with what I call endogenous viruses, some viruses that have been integrated in our genome for the last 25 million years or so. And that there's evidence that they are mostly repressed, but they could be woken up under certain circumstances, like with aging, for example. Okay, so I'm fascinated by that possibility. And so if I had means at hand, this is, in fact, we've started looking a little bit on this in the lab initially, but this is what I would do.
Oh, this is very interesting. Well, thank you for sharing some of these projects, these dream ideas that you'd love to research more that maybe keep you up at night. I think these are wonderful for me and our listeners to ponder through our day and expand our own minds. So I appreciate that. And we've covered a lot of ground, but Sandra, I'd love to end our conversation talking about advice. So when you think back on your own career, is there a particular piece of advice that you received that really helped you that you can pass on to our listeners out there today?
It's about decision making and how we always throughout our lives, of course, at every step, we have to make decisions. And one of those came about when I had the fortune of having choices to make where to start my lab as a faculty member. And so I did my due diligence, getting the pros and cons and everything. And I investigated everything and thought about everything. And I was a bit stuck. So I asked a colleague, a class colleague and friend, for some advice. And she said, well, you've done your homework and you know already where you want to go. It's really a gut feeling at the end of that after you've done your homework. But whatever you do, just go for it and don't look back. And I think that advice has really served me well, because taking up this job is a challenge. It's not necessarily for the faint of heart, in the sense that we have to fight a lot for funding and publishing in the careers of our mentees that we are very much involved with. But the advice of don't look back and just keep going is what I go back to a lot. And then inspired by all the other scientists that we have talked about earlier, is just keep fighting the good fight, which is a theme that comes back often because we encounter a lot of obstacles in this career. And so I think that's the advice that I'd give forward, is just don't look back and try to fight for what you think is the great science that you produce.
Well, that is excellent advice for our listeners out there. And I'm really glad you mentioned it. I think kind of thinking about all these what ifs, these alternative scenarios, if you had made a decision differently, can be really frustrating and demoralizing and not productive ultimately. So I think continuing to look forward is so important. So wonderful lesson to impart with our listeners there. Is there any other last piece of advice or a last note of inspiration that you would like to leave them with today?
The inspiration that I get in the science is really, I think about the people that do the science where I'm mostly inspired. And so I think that if we embrace the people that do the science, then we're doing science well. So this is our amazing students, the postdoctoral fellows, technicians, the research specialists, our assistants, and our colleagues that do the science. It's where it is. If we embrace them, take care of them, encourage the diversity in their thoughts, in their beings, then I think the science will follow. So I think this is our legacy. The legacy that I can leave is in the people that do the science and the science that comes from that process of embracing the people.
Well, Sandra, we couldn't agree more that it is all about the people behind the science. So a fantastic note to end on there. If our listeners are intrigued, Sandra, and they want to learn more about you and the excellent work that you're doing, what is the best way for them to do so?
So happy to be contacted. We have a website. People can look us up and see our interests. They're highlighted there a little bit more in detail. And I'm also excited to hear from anyone with comments or questions via email. And also if anybody wants to join our team. So email is great. That's the best way to contact us, I think, and to contact me.
Phenomenal. Well, listeners, definitely check out Sandra's website. Get in touch if you have questions. And Sandra, it has been such a pleasure to chat with you today. Thank you so much for your time.
Thank you, Marie. It's been really wonderful. Thank you so much.
Well, we've enjoyed this conversation. Listeners, it's been great to have you here with us as well. And we hope to see you on our next episode of People Behind the Science.